We'd now like to move on to highlights from the Research Networks and the Data Processing Analysis and Coordinating Center, which I'm going to call DPAC, the whole rest of the meeting here. And we're going to have three presenters today. The first is going to be Dr. Carrie Bearden from the ProNET network, who is at the University of California, Los Angeles. Dr. Barnaby Nelson, who is the lead on the Prescient Research Network and from University of Melbourne in Oregon. And then Dr. Martha Shenton, who is the lead on the PREDICT DPAC and from Brigham and Women's and Harvard Medical School. So turn it over to you, Dr. Bearden.
Thank you, Linda. Can everyone hear me okay? Excellent. And see the slides showing full screen? Okay, fantastic. Well, since I'm already 9 minutes behind schedule, I'm going to jump right into this. So welcome everyone to the second, I believe, annual meeting of AMP Schizophrenia. So I'm Carrie Bearden, as Dr. Brady said, representing the Psychosis-Risk Outcomes Network. Here are our sites involved. And so just to jump right into the issue at hand, there is a critical need for early intervention for serious mental illness. And from this meta-analysis from one of our ProNET collaborators, Paolo Fusarpoli and colleagues, we can see that the rate of clinical high risk for psychosis in the general population is a little under 2%. And in a clinical population, a help-seeking population, it's about 10 times higher. It's 19%. So there's really a strong need for interventions for this clinical high risk state. And while the clinical high risk state for psychosis is increasingly recognized as a valid diagnosis associated with serious mental health outcomes, and we now have specialized clinics for clinical high risk youth, for adolescents and young adults, increasingly worldwide, but there are really still no registered medication treatments for clinical high risk for psychosis. And part of the reason is that there are some really key questions that are needed to design trials that are that actually lead to significant public health benefit. And some of those key questions, which we are hoping to address with our network, what are the key outcomes that could potentially lead to treatment indications? What measures should be used to record these outcomes? And then what is the optimal trial duration and measurement schedule for each outcome? And then finally, how should these clinical high-risk patients be selected for each outcome? As we know, it's a very heterogeneous condition. And so we have two harmonized networks. We started out, we had two completely separate grants that were funded. And so we had to make it all into one project, which has been really fun. Has been really fun, seriously, working with all these collaborators. So the goal is 1,977 clinical high risk subjects across the two networks, ages 12 to 30, 42 international sites enrolled over 2 years, 1,000, close to 1,000 within each network. And so our network, ProNET, has 26 sites across North America, Europe, and Asia. And you'll hear from my colleague, Dr. Barnaby Nelson, next about Prescient. Participants are followed for 2 years with identical measurement protocols with outcomes and biomarkers that have been really carefully justified. And then we're also, and this is really critical for assessing normal developmental change within these measures, 640 demographically matched healthy controls. So some of the deliverables from this project, a panel of outcomes that could potentially, well, we hope, will lead to new treatment indications, for example, and for some of these dimensional symptoms. So example, for the treatment of cognitive impairment in clinical high risk patients. Recommended measures to record these outcomes and strong estimates of their expected values and variability over time in future clinical trials. This is really critical, as well as recommendations for trial duration and the measurement schedule for each outcome. And one of the innovations that I'm really excited about as part of this project is that we'll have both static, as in baseline, and as well as combined static and dynamic subject selection tools that will optimize stratification of clinical high-risk individuals for each of these outcomes. And then lastly, I think this is something we all should be really excited about. It'll be a publicly available landmark data set. This is really a big change that's really different from what's been done before in this space, that it's going to be a publicly available resource that will provide a solid guide to the next generation of research and public health advances in this field. So I just want to give a quick shout out to all of our really fantastic ProNET core directors, everyone that's part of these teams that have just been doing an absolutely incredible job. Just briefly, Jean Addington on the clinical side, William Stone, Bill on the cognition, Paolo Fusarpoli on envirom. Philip Wolf is leading natural language, John Torres on digital phenotyping, Mike Harms, and as part of one of our neuroimaging team leads, Alan Antichevich on data transfer, Dan Mathelon, EEG, Deanna Perkins on genetics and fluid biomarkers, and then Ty Cannon is our data analysis liaison, and Ty also led our previous Naples North American Prodromal Consortium. And then here's all of us as the multiple PIs, Scott Woods, John Kane, and you can see we're all looking, so super young and happy, and no, we're just kidding, we're still happy. So our sites, I don't have time to go through everyone's names, but these are all of the sites and PIs who all have really incredible teams that have been working really hard to prepare for this. So as I mentioned, one of the challenges was putting together these two networks. And so one of the deliverables, is that we integrated, this was done by Scott Woods, Melissa Kerr, Barbara Walsh, Allison Young, colleagues. So between the Prescient and ProNET networks, the Psychs, this is a new instrument, integrated. It stands for Positive Symptoms and Diagnostic Criteria for the CARMs, harmonized with the SIPs. So these are the two assessment measures for clinical high risk for psychosis that we integrated into one. They've developed new training materials, new videos. This is really, I think, a major deliverable that we now have one integrated assessment measure for the clinical high risk state. I'm part of the neuroimaging team, so I'm gonna highlight some of the neuroimaging innovations, which I'm really excited about. So we're using this pretty cutting edge imaging technology developed for the human connectome, which allows reconstruction of brain connectomes at multiple spatial and temporal, really exquisite resolution. And so just here, these are data from Yale. Thanks to Alan for sharing these with me, showing just across these different modalities of imaging, the quality of data. This is showing data acquired from 2 subjects over 2 days. So the top is our structural T1 and T2 weighted acquisitions. The individual, those free surfer segmentations you can get from these. This is a T1, T2 map ratio for myelin mapping. This is for resting state functional connectivity, surface-based fold. And then this here is a seed-based analysis from our resting state scan. And then on the bottom is a probabilistic tractography from our diffusion weighted imaging sequence. And so this is, I think, a really exciting innovation for the project. This is our scanning certification tracker. So one of the nice things is that the vast majority of sites have Siemens platforms in the ProNET network. Most of them are Prismas, and there are 3 Skyras as well. So the vast majority of sites that can be certified for scanning have been. We're still almost done with the GE protocol. And I want to just shout out to Mike Harms and Ofer Pasternak, our cross-network imaging leads, who have developed some really great training materials and a rigorous certification process. So just quickly, I'll show you what our, oh good, I still have 1 minute. What the protocol looks like. It's a very time-intensive protocol for participants to go across all of these different biomarkers. And I just want to highlight a couple things here, which is that the biological, the biomarker assessments, imaging, EEG, and blood and fluid biomarkers these are all done very close together at the early phase, baseline and 2-month follow-up. Other assessments are ongoing throughout the 2 years protocol. But the rationale for the 2-month repeat biomarker assessment is based on our preliminary analysis of existing data showing that change over this time period can be really useful for prediction. And so I just want to, in my last minute, 10 seconds, highlight the value of, you may think that 2 months is not long enough to show meaningful change, but these are data from Megan Collins, Ty Cannon from our Naples consortium, really showing that there is significant change over time in those who who subsequently convert to psychosis. So this is prior to the onset of full-blown illness. We're seeing a difference in trajectory. So this cortical thinning really does show promise as an early biomarker. The multimodal data collection that we're getting is also going to be, I think, really helpful for potentially seeing tipping points in high-risk young people with our remote monitoring and digital phenotyping. And lastly, we've launched, yahoo! So we have 3 participants that have been scheduled at Yale, and then the 3 other sites that are just waiting for a green light to start. So I'm thanking our participants and families and all of our partners, and I'm done.
Thank you very much, Dr. Bearden.